Okay, recording in progress. Pooja, no sir.
Yeah.
Yeah.
Okay. Hi guys. So today we are, okay, my name is Pooja
I'm Amy.
and we are your supportive TAs. And today we will go over topic 7, overview of cell cycle in cancer biology. I really like this topic, this cell cycle in cancer biology, because I'm very interested in cancer research. I don't want to do research, but I'm just interested in cancer. So we're gonna go over what does cell cycle refer to. Do you guys know? Any idea what cell cycle is?
The process at which the cell divides and makes two daughter cells.
So it's like the process of G1 phase. It checks if everything is everything is compact and is put together, and if there's anything is wrong in DNA, then it will either like go to what cell death, or it's gonna process if it's everything is normally functioned. So a time during which the cell grows, replicate DNA and divides. That's what cell cycle refers to. A time during which the cell cycle grows, replicates the DNA and divides. So in this topic, we're gonna discuss about eukaryotic cell division must be controlled carefully because cell division is multiple step copying the DNA, and separating DNA is complicated because of the consequences, mutation that it can lead to, or it can happen in the process. There are many things that can cause cell division to divide incorrectly. This can lead to
Apoptosis.
apoptosis or over
Proliferation.
Yes. Sorry, I'm bad at pronunciation sometimes. So that means like cell death. Transition into cell cycle. If I'm going too fast, just let me know and I can slow down. Okay? But we will send you the slide after. So if you just want to listen and take the notes after, that's fine too. So cell must meet certain condition in order to divide, right? Do you guys know what those processes are and the circumstances and conditions? Yeah. Yes.
Like no damage.
Yes. Correct. Don't want to divide if there are not enough resources, just like how you said, if there's not enough nutrients, if the cell is not big enough, then do we want to go and divide? No, right? So it makes sure that everything is fine. We have enough nutrients and cell is nicely and healthy for cell division to happen. So number one is growth factor signal. Second, nutrients, carbon, nitrogen, and other factors. And we talk a lot about ATP, which is energy source that we need in our body and in our cell. Regulatory proteins. Without proteins, we can't function it. And then now we're gonna talk overview of the cell cycle and go over the whole phase of cell cycle and the checkpoints. Okay, so we have G0 phase. I'll read off what this is and we can briefly think what you guys know so far. So this is the default state of the cell, not technically part of the cell cycle to leave G0. The cell must receive a growth signal, right? So that's G0. Fully indifferentiated cells never leave this phase. Okay, G1 phase. This is where we make sure that DNA is being replicated and we're preparing for replication. So G1 phase, growth 1, or phase 1, the cell prepares for replication of DNA. S phase synthesizes during this phase, DNA is replicated, synthesized. To begin this phase, a checkpoint is needed. G2 phase, right, which is called growth, phase 2, the cell communicates to grow and prepare for M phase after replication. So if you can look at the image, it kind of goes from like G0, G1, and then G1 checkpoint. And here, if you can see, it just goes S phase DNA replication, and then it goes back to like this whole phase, if you can see there. And those are the checkpoints. G1 is like checkpoint. Just make sure that everything is put together compact. And then M phase is where separation of chromosome happens and division of cell. Of course. So like we know that we have 2 chromosomes, and this is where the separation of chromosome. You want to tell us?
Oh yeah. Okay. So now we're going to talk about CDK, which is cyclin dependent kinase, and what do kinases do? Yeah? Yeah, they phosphorylate things. So CDK's primary function is to phosphorylate things when it's bound to cyclin. So it's like a switch of the cell, and it allows the cell to progress through the cell cycle depending on what type of cyclin is around. So everyone knows that there's different types of cyclin, right? Yeah, so there's different types of cyclin, and they're produced at different times of the cell cycle. So that will help the cell know like what parts it should go through, if that makes sense, like which checkpoints it should pass through at certain times. And that's why the regulation of CDK is very important to pay attention to. 3 regulators of CDK are cyclin, which we just talked about, right? So CDK binds to cyclin, and that allows it to get phosphorylated and activate its activity. It's also regulated by 2 kinases, CAK, which is cyclin activating kinase, right? So that one phosphorylates cyclin, or not cyclin, that one phosphorylates CDK so that it can go and phosphorylate other things. And then there's another kinase that phosphorylates CDK, which is WEE1, and that's an inhibitory kinase. So when it phosphorylates CDK, WEE1 actually stops the activity of CDK from happening until it's necessary to happen. So it's like dual regulation so that things don't happen too quickly or too slowly. And they work together to double phosphorylate CDK. And then there's also other protein inhibitors which can be introduced to the cell when you want to like upregulate CDK or downregulate CDK if there's a mutation, like a loss of function or a gain of function mutation, and those can be related to cancer and how you can like research cancer or cure cancer or things like that, or regulate the cell cycle if there's something going wrong. So what are ways that cyclin, a protein, can be regulated by the cell at different times? So can someone think of an example, someone who hasn't participated?
Well, outside.
Yeah, of course. WEE1 is a kinase. It's a type of kinase that phosphorylates CDK.
What's its name?
I actually have no idea. I think that's just the name.
I will actually.
You know that? Yeah.
It's actually kind of a funny story. WEE1 was discovered by an Irish guy, and it's called Wee as in little. Oh, that's actually the name.
That's what I was thinking. I was like, it just sounds like little one, like WEE1, like slang.
Mm.
That's fun. I like that. That's cool. Can anyone else think of an example now? Yeah, I think everyone's just like tired. That's okay. So just different ways that cyclin could be regulated is if you introduce a protein that like a protein. I feel like I answered my own question earlier already. I don't know why I did that. I'm sorry. I think I wrote that and then I wrote the information on the slide later without seeing that I had that question there. So we're gonna move on. Did you want to read this one?
Yeah.
Okay.
So now we're going to talk about the CDK and the G1 checkpoint. So the checkpoints are where it makes sure that everything is properly connected and is functioning to move on to another phase, okay? So G1 this is a diagram, okay? So if you look here, okay, cursor is working. So if you look here, this is like the growth factors where either it's going to go to cyclin and then it will go bind to CDK, then it will phosphorylate. And once they phosphorylate, then RB is gonna activate and then it will use the ATP. Okay. So then growth factor arrives from other cells. And growth factors causes the increase in cyclin in E2F factors, which is the concentration, and it's basically like transcription factors. And cyclin factors So this is the process of like where cycling factors bind to CDK. CDK then gets phosphorylated in the RB, which inactivates the E2F by binding to it. CDK, once CDK is activated, then it will dephosphorylate the catalyze, which it catalyzes the phosphorylation of RB. Then the RB releases the E2F, and then E2F here, if you can see, once it gets released from the RB, the E2F enters the nucleus and triggers the production of the S phase, which is here, the
like
G1 checkpoints.
I'd like to add something.
Mhm.
I put a typo on the slides, and before I upload them, I will fix this. But where I wrote here that it says RB's active mechanism is to initiate S phase, that's wrong. RB gets phosphorylated, and because when it's phosphorylated, it releases E2F, whose active mechanism is to initiate S phase. So
Yeah.
that was a small typo. I'm so sorry, guys. I'll fix it later.
Want to add something? So
Yeah.
see how you have the two phosphors here and here? So the bottom one comes from CAC, which is the activating enzyme to activate CDK, and the top one is CDK, and the top one comes from WEE1, and which is the inhibitory enzyme.
Yeah.
Mhm.
So just so.
So there's no confusion.
Yeah.
So then
Yeah.
once
No,
the
he has a question.
oh sorry.
So RB, does it release EF2?
It does.
It does.
It does release EF.
RB does release the EF2, and then EF2 will go to nucleus, and then it triggers the production of S phase production.
And I'll fix
Yeah.
that when I upload slides.
So once RB is activated, it will release E2F, which will go into the nucleus and make transcription factor.
Yeah.
And
E2F
yeah.
is a transcription
So
factor. Yeah.
it's actually technically when it gets phosphorylated, it becomes inactive.
Yeah.
The RB.
Yeah.
So RB is always active,
Yeah.
holding on to E2F.
Yeah.
The phosphorylation basically changes, it's basically like two hands holding the E2F.
Yeah.
The phosphorylated hands open, it releases.
Okay.
that.
Yeah.
And so that's basically, there is an affinity.
Mhm.
Yeah.
Okay. Basically, see when RB is active or activated, it's actually become inactive.
Yes, it's being, I would say it's getting inactive.
Yeah, it's getting deactivated by getting phosphorylated.
Yeah.
So phosphorylation leads to its inactive state, and I'll update all of that so you can reference it later.
Okay.
Yeah.
So once it does its job, it becomes inactivated.
Yeah.
Yeah. So then once the cell is ready, it will go to CDK, then it gets dephosphorylated, which leads to activation. And CDK activates the mechanism of RB, basically what we talked about there. And then these are the checkpoints. If the cells do not commit, they pass respiration.
Oh, if the cells do commit, then they pass the restriction. There's supposed to be a C there.
Okay.
I'm sorry.
It's okay. They pass the restriction point into S phase. Before this point, they can still return to G0 phase and not undergo
Apoptosis.
apoptosis. So they can return back, and it will make sure that it doesn't die off.
So it can only change its mind in the G1 and G0 phase, essentially.
G0.
During the G1 phase, it can change its mind and leave the cell cycle, and G0 is not part of the cell cycle.
Yeah.
Okay. So after G1,
So basically, after E2F is unbound, you can't go back.
Yeah.
Mhm.
Checkpoint occur like G1 phase will occur, then there's the checkpoint, and then S phase there's a checkpoint.
There's checkpoint in
It's-
G1 phase.
Yeah, the checkpoint is kind of at the very end of the G1 phase. So basically, what would happen is that as the growth factors continue, then like as the cell senses that it's ready to move on to the S phase, that will trigger the dephosphorylation of CDK, which is a kind of relatively gradual process. It's not like instantaneous. It's not like the cell decides, okay, right now at this very second I'm in S phase, but it's like at that time, that's the G1 checkpoint. As it passes that checkpoint, it then starts releasing E2F, and once E2F starts activating transcription in the nucleus, then the S phase that's like when the S phase has started.
So there is after the G1, there's gonna be G1 checkpoint just to make sure that everything is properly functioning. And if there's like any mutation that occurs, it will lead to cell death because you don't want that because it's just gonna lead to like an inhibition to other proteins. Okay.
Okay, so the S phase is not actually that complicated. For the purpose of this class, you don't really need to know that much. This is DNA replication. Don't worry about that diagram. I just wanted to show you what DNA replication kind of looks like. But basically, DNA replication occurs, and you've got all these proteins working to make a second copy of DNA. And what really you do need to know for this class is the right side of the screen, which is where the centromeres and the homologous chromosomes get duplicated, and then you have two sets of homologous chromosomes. And so, in conclusion, S phase just produces a second template, which is the DNA. Yeah.
No, wait,
Unless you want me to talk more.
no, you can do.
Bye. Okay, so this is G2 and the G2 checkpoint. It's honestly not that complicated either. This is just growth phase 2. It creates the proteins needed for M phase, so proteins that would make up like the spindles, right? And proteins that make up the what are the ones that hold them together?
What is it? Centromeres,
The protein, the
kinetochore,
connectochore, yeah.
the kinetochore.
Any proteins that might be involved during the M phase that the cell wouldn't just naturally have lying around for other things, right? They're going to want to produce those proteins for the M phase. So that's what they're going to do during G2. And then they're also going to check for DNA mutations, damages, and breaks. So basically, if the DNA is just wrong or it's not where it's supposed to be or it got duplicated incorrectly, because there's a lot of things that can go wrong during the duplication of DNA. It just fixes all of those things then so that each daughter cell gets a healthy, full copy of DNA. Yeah. And so the G2 checkpoint basically just makes sure those two things that happen during G2 goes correctly. It's looking for undamaged DNA and the full synthesis of the necessary M phase proteins. So then once it passes that point, it can move on to M phase. And I don't believe that for the purposes of this class, you really need to know anything about the mechanism of movement into the M phase. Right? So you just need to know what you need in order to move to M phase. You don't need to know how it moves into M phase. Yeah. Any questions? Right. So this is a little bit more about the M phase and the spindle assembly checkpoint. The spindles are really doing the most important thing during the M phase, which is physically separating the chromosomes. So if you have your two chromosomes, they're gently pulling them apart so that each new cell gets a copy of the genetic information, which is very important. And you'll actually talk about that later, what happens when it goes incorrectly and the different outcomes of that. But for now, all you need to know is that the cell is separating them and it wants it to go well. So that's what we're looking at here, the spindle assembly checkpoint. So we're going to work backwards for this one. So if you see over here where it says metaphase, right? You have cohesin, which is actually the one I was thinking about earlier that I couldn't remember. You have cohesin, which is holding your identical copies together, right? And there's like a bunch of these. So this is just like one picture of one of them, but you've actually got a bunch of them all held together by cohesin lined up in the middle of the cell, right? Yeah. So then you've got this, but what you want in order to pull them apart is to break cohesin, right? Right? So in order to separate them, you need to like have them not bound together. So they actually get broken up by separase, which is an enzyme, right? Yeah, it's an enzyme. So separase has to be active though, and you don't want separase going around and breaking cohesin randomly until it's time for that, until everything has lined up at the center of the cell, right? And everything is ready. Because yeah.
I'm sorry. Cohesin, what is that? What is that binding? You're just talking about breaking it.
Yeah.
What is it bound to?
So cohesin is basically it's a complex, and if I remember correctly, it's actually, and you don't need to know this, but just some context, it's added in S phase to hold the chromosomes together.
Oh, so that's what's holding.
That's what, yeah, so they don't separate because every cell needs to have the right amount of chromosomes. Otherwise, I mean, that's where we get, Down syndrome, stuff like that. That's where we get those kind of issues is when we have too many or too little chromosomes.
Right.
So basically it's holding them together, and it is actually the way the cell tests, because the cell can't see, and she mentioned this in class today, cell can't see how the, like, where it's reaching with the, what are they called again?
Oh, the spindle?
Yeah, the spindles. They can't really see where
Microtubules don't have eyes.
yeah, they don't have eyes, so they end up connecting to this, and it's actually the tension of each, I believe, centromere on either side, pulling on either side of them that tells the cell it has the right kind of lineup. And that's why you see these things like in bunches here. Is it's technically, yeah, so you've got the green ones that are pulling from either side. And so when the cell sees that, feels that, that is actually the end phase checkpoint, because it knows like, hey, we're lined up. And then it goes back to this. So that, clear that up.
That's really cool. I did not know that. Thank you. Did everyone hear that? Yeah? All right, good. That was really cool. I like that. So then separates, goes, and it cleaves cohesin, so that after all of those things Ben just described, the cell can separate the chromosomes and each daughter cell can get one set of DNA, right? So in order to do that, separase has to be active, but you don't want it to be active too soon, which is why you have an inhibitory protein called securin, which, as I described it to my friend when I took this class, it's like holding the hand of a child that really wants to go and get like a sweet treat at the fair. You're just holding on to it until it can go and run and do its thing, right? But you want to hold its hand until then, until the right time, until you're like, yep, you've been good, you deserve a treat. But that's kind of weird. So basically, securin is just holding on to separase and stopping it from being active. And then you have this other enzyme that is APC that comes and tags securin for degradation. And then your enzyme separase can go and cleave cohesin so that they can be separated. Does that make sense? Yeah. Any questions? Okay, good. You?
Right. Cancer. We all love to talk about cancer. So basically, there are in the body, there are two aspects that can go wrong. You've got tumor suppressors and oncogenes. So the tumor suppressors are like P53. So basically what P53 says, when you have DNA damage, it goes, all right, stop the show. We're not doing this. And it stops things. That's why it's a suppressor, because it stops stuff. Where proto-oncogenes are, what is it? E2F.
E2F is a
So E2F is a proto-oncogene because if E2F is always released, it's going to make the cell go wee and just keep on doing whatever it's doing and producing. So that's just keep that in mind, because that's two very important concepts. Okay. So and the analogy here is the tumor suppressors are the brakes. They're like, nope, buddy, don't do that. And then the proto-oncogenes are the accelerator. We're gonna go for it.
Yeah. So you all know, like you kind of sit there and the car doesn't go, but then you have to have some acceleration to get the car to go somewhere, so you go where you want. But then you also want to have brakes so you don't hit other people on the road. So that's the importance of those.
So I guess we're going to go a little bit into P53. So basically P53, it's part of the process. You know, there are so many details to this, but it's part of the process that if you have DNA damage, P53 will get phosphorylated and become active, and it will put the brakes on and say, okay, pause. Can we repair this DNA damage? So that's the pause cycle, and it upregulates P21 CDK inhibitor, which is actually part of putting on the brakes. There are other components that try to repair the cell damage and correct things, which you don't need to know. And so if it stays in that phase long enough, it will then signal programmed cell death, apoptosis. So if there's a disruption of P53, it'll lead to uncorrected DNA, and that's where you get, that's what cancer is you're getting mutations. Make sense? Cool.
Oh, we already said that.
Yeah. I tend to just get out of myself. Okay, so there's promotes, so proto-oncogenes yeah, I guess we kind of already talked about
Yeah.
this. Promote cell growth, oncogenes, gain of function, produce a lot of the cancer cells. So it's not really,
The point of the slide is just that oncogenes are not different genes than proto-oncogenes. They're just upregulated proto-oncogenes, whereas tumor suppressor genes are different. Those are like different, those are different proteins, different regulators, they're doing different things. But oncogenes are just like a gain of function in a proto-oncogene. If that makes
Okay.
sense.
it's like, what are we saying in this slide?
It's because I don't write that much on my slides. I put it on my brain and then it's in the speaker notes.
So, over proliferation. So again, this is a proto-oncogene. So mutated RAS, the mutated RAS gene. Do you guys remember the RAS pathway? So that is an example of a proto-oncogene because it'll just create, not what we want. So, and that's because it helps with cell growth. So I guess I kind of went through this, goes nonstop. It's like the gas pedal stuck down. All right, so P53, we talked about this before. If we don't have it, we don't have that stop function. So you're not going to get when there's cell damage, it's not gonna stop in G1 or G2 checkpoint. It's not gonna be able to stop for that reason. Now, I think there are other mechanisms, but this is one of the main mechanisms, and because each mechanism targets a certain component, and this is the one we're talking about, so it's the one you need to know. So again, it stops cell damage when if it notices DNA damage, it stops cell replication if there's DNA damage. And so, like I said before, and then too much cyclin or CDK can make the cycle go too fast, which is basically, think about a cell. A cell is like a large place. If you increase your amount of CDK, you're going to get more activation of something. And so if you get too much activation of something, and the cell is very much an alive place, you don't have one CDK and one cyclin floating around. You actually have a lot of these things, and it's about concentrations. And so if you have really high concentrations, something's probably gone wrong in the cell. But even to regulate that, the cell will always has things that are ubiquitinating or basically changing the conformation of these things so they're not activated. So it's kind of always a battle in there. Is there anything you wanted to add to that? All right. That's what we have.
Does anyone have any questions?
Yeah, we can go over and cover if there are certain, if there are certain areas that anybody wants to cover or has questions on.
Yeah, I think that's very specific questions.
So, I'm confused why CDK, has two, like, I don't know what they're called, so like, I'm confused why it has like CAK and WEE1, like, so CAK is, I know, activating CDK, and WEE1 is inhibiting it.
Yes.
So like, so what's the point, like, I'm confused.
So those are all, remember like the RAS MAP pathway? So those are enzymes that are part of different pathways.
Okay.
And so WEE1 inhibits CDK until the cell's ready to move forward.
Okay.
So a couple slides, let me go back to that slide if I can find it. So see how on this diagram you have the two phosphoruses here, or it's been phosphorylated twice. One of these, I believe it's the upper one, but again, they are always changing shape and who knows how they're presenting this. But one of them is from WEE1 and other one is from, I think is what you were talking about. It's actually K. And so the cell doesn't want things, it wants things to happen at a very specific time. It's like the example of driving a car. When there's a stoplight, you don't want to hit the accelerator. And so this is just holding it, the phosphorylation by WEE1 is holding it inactive in doing its job because CDK goes off and phosphorylates other things itself.
Right.
And you can say, the way I like to think about cyclin is it's actually a pair of eyes. Cyclin tells CDK where to go and what parts of the process to phosphorylate. And so that's why CDK is used throughout the entire replication cycle. Is it's just its only thing is, oh, I'm gonna phosphorylate whatever I get put in front of me. It's cyclin that tells it where to go.
So cyclin, the point of that being bound is that it will tell it when to go.
Yes.
And then CAK, the activating or CAC.
Yes.
That will tell it when it's supposed to help divide. And then V1 tells it don't divide yet.
So
Can I?
yeah.
Okay. How I like to think about it is like, what does phosphorylation do in like the signal pathways? Like, how does phosphorylation change a protein when you get phosphorylated? Yeah, it changes the function. So whatever the phosphate group or it's a phosphate group, right? Yeah.
Yeah.
So whatever the phosphate group here is doing is just changing the function so that it's not doing anything. So it's inactive, so it can't bind to other molecules. Maybe it's like attracting another amino acid, so it changes, so it's like more closed and you can't like go and bind an active site. Maybe. Like that's a hypothetical. I actually have no idea. I haven't really looked into that. But and this phosphate group is changing its conformation so it can go and bind to other things. Maybe the thing it wants to bind to, like, is really positive, so it needs to be more attractive in that place. Again, that's really hypothetical. But both of them are just changing its function in a different way so that it does different things. So then if you remove one of these, it changes its function again. And so now with one phosphate, it's active. With zero phosphates, I think it's probably inactive. Yeah,
Yes.
with zero phosphates, it's inactive. With one phosphate, it is active. And with two phosphates, it is again inactive. And I would just think about it, just like, give yourself like a hypothetical example that works for you, because it's just conceptually understanding that having another phosphate group is just changing its shape again so that it can't do what it would be doing if it had one phosphate group.
And she did mention that in class, that phosphate, I think, basically puts like a nub in the way of CDK's binding site, so it can't bind what it wants to bind.
Yeah.
Now, I also, in your question, I heard a different kind of confusion. So this right here, this is the cycle for the CDK in the G1 checkpoint. CDK does a lot of many other things besides this. This is just one example of, because this is whatever, I think it's maybe G cyclin. I'm not sure which cyclin it is.
It might
The
be.
G cyclin.
It's probably G1 cyclin or G1S cyclin.
So and due to that, because this is actually technically G1 cyclin, it's telling CDK to do this specific example. CDK does lots of other things and has lots of other aspects within G2, it's the thing that moves the entire cycle forward. But it has a lot of other jobs that we won't discuss and you wouldn't discuss until upper level courses.
Yeah.
Probably could turn that.
Yeah. I'm gonna turn off the recording now. So if anyone watching the lecture online has questions, just email us, but that's about it, and we'll upload the slides.